MIT acknowledges indigenous peoples as the traditional stewards of the land, and the enduring relationship that exists between them and their traditional territories. The land on which we sit is the traditional unceded territory of the Wampanoag Nation. We acknowledge the painful history of genocide and forced occupation of their territory, and we honor and respect the many diverse indigenous people connected to this land, on which we gather from time immemorial. We'd now like to give a quick run-through of the structure of the webinar today. We'll begin with a quick introduction to the definition of biomaking and how that's shaped the community we aim to create at MIT. We'll then introduce the seminar series and finally open the door to Devora and Najjar. And a quick note about logistics, please enter your questions in the Q&A box, and at the end we'll have a bit of time where attendees can raise their hand to speak. And also note, this webinar is being recorded, although the attendees are not in the video, and will be released to the public.
So what exactly is biomaking and why are we called the Biomakers? At least to us, biomaking means a lot of different things, but particularly for this seminar, we'd like to highlight that there is an important community aspect of biomaking. And there is a high importance of inclusion of different sorts of people at the table when it comes to what we make and what we do. And so beyond that, we want to support a lot of student-driven projects and interdisciplinary research, because we believe that it contributes to this community. And we aim to involve the public, and that's kind of the ultimate goal of making biotech and biology as a whole. So who are the MIT Biomakers? We are a student club that contributes to the maintenance of the Huang Hobbs Biomaker space. A lot at the very core of like why we exist is to create community around this space. And this is a community of mostly bio-curious people, not necessarily biologists or chemists. Some of us are just makers that want to bring biology into what we do. So we help foster student-driven work to this community, and we're graciously supported by the Huang Hobbs Biomaker space itself through its resources, through the faculty that support it. If you want to find more information about our space, feel free to visit our website, biomakers.mit.edu.
All right, so a little bit about the speaker series. In this series, we've invited a range of speakers who are leading biotech companies, nonprofits, and citizen scientists looking toward making the realm of biomaking for social good more accessible. In addition to her contributions in research, Dvorah was invited to speak today because of her experience for taking into account social context of the work she does. In this series, we would like to promote biomaking organizations where biotechnology is developed and used to solve local and global problems that everyday citizens and people face.
Yeah, so before we begin, we'd like to further define equity and social action, just to clarify, clarify the meaning of equity and social action as we're talking about it. Often equity gets conflated with equality, which means providing the same to all, but we're here to define equity as fair treatment, access, and the opportunity for all people to recognize that we don't all start at the same place. So to that end, we'd like to highlight several questions on this slide that we aim to bring to the forefront of this series. And on that note, we'll introduce our speaker of today.
Devora Najjar is a PhD student in a Collins lab and a media lab in the Responsive Environments group. She graduated with a degree in chemical engineering from the Cooper Union in 2016. And now at MIT, she's focusing on paper-based biosensors for ecological monitoring. She is additionally interested in equitable dialogue on biotechnology policy, biosecurity, and environmental management for a more diverse conversation. She has also helped design neuro-inclusive educational programming in ecology, engineering, and evolution for students in the Boston area. Please feel free to add anything to your introduction as well, and the floor is now yours, Devora.
Yeah, so I'll stop sharing on my slide. I believe you do have permissions to share screen, but do let us know if you have any technical troubles with that. But thanks for.
Sure. Thank you, guys. Okay. Let's see. Share this one and then present. How does that look? Well, it's loading, but can you see it?
Looks great.
Okay, well, hi everyone. Thank you so much for having me. I'm hoping this could be more of a conversation than a presentation because it's always great to hear about different people's ideas. I am really excited to talk about this topic. It's not something that I've maybe presented in a very formal fashion. A lot of the community engagement work that I do, it's really just me with the communities, and I'm still trying to find more academic spaces to speak about it. I think the more you do it, the more you feel like it's not ready to be presented yet, but that's okay. So like you heard, I'm Dvorah. I got my degree in chemical engineering from Cooper Union. I did my master's at the Media Lab at a group called Sculpting Evolution, and now I'm doing my PhD with a group called Responsive Environments. And I guess just to explain a bit about my trajectory in grad school, which was a bit non-traditional, I started out in a group called Sculpting Evolution, which focuses on gene drives. I was quite interested in this sort of intersection of biotechnology, synthetic biology, and people. And gene drives seemed to be like one of the really big things that sort of forced the conversation. Gene drives is genetic engineering that sort of spreads in the wild through biasing inheritance for specific traits. And so I was working on it in particular for trying to figure out if we can more sort of humanely and biologically get rid of invasive species. And this is just like a screenshot of one of the papers that I wrote while I was in the lab at the time. And I think, you know, it's funny to look back at this stuff and see that the same through lines are still present in a lot of the work that I do now. But doing this gene drive research was my first experience in working with communities where we would talk to people who were dealing with a lot of invasive species issues and thinking about how could we prioritize your community's needs, and also thinking about the types of risks that are inherent in adopting a new technology that hasn't really been tested before, and thinking about how you can sort of do that in a way that's ethical. And I feel really lucky that MIT has such a great infrastructure for technology policy, and I took a lot of classes thinking about what it means to do sort of the policy side of things and the risk assessment side of biology. While, like, you know, during the day I'm in the wet lab, but sort of at night, that's the kind of reading that I was doing. And it dawned on me that there's still so much that we haven't accomplished within biological monitoring of our environments. And it seemed almost crazy for me to be jumping the gun so much and thinking about genetically altering our shared environment before we even have a sense of what that environment is. So I kind of backed up a bit and I switched directions for my PhD, and I focus now more on ecological monitoring tools that we can be using that implement biological engineering, but as a way to figure out how we can provide more tools to people who are non-scientists in the outside of lab context for environmental monitoring, mostly so that communities have a better sense of what's going on within their own environment, and also so that, you know, they can be maybe a precursor for whether or not communities want to think about adopting biotechnologies in the future as our environments continue to shift due to things like climate change. And so this is one of the pictures. So I've done a whole range of sort of engagement work, and I didn't really show most of the pictures, both a lot of the time because it's a lot of children and it feels like really exploitative to show lots of pictures of children, even if I get their, you know, parents to sign waivers and things. But this is a drawing that was done by one of the students I worked with in Hong Kong, actually talking about what it looks like to study urban water bodies, and that was through MIT's PK through 12 STEAM program, which is great. And shout out to all the people there who have lots of time and resources if anyone's interested in figuring out how to do more teaching. But we sort of asked the students to think about what encompasses the ocean, and thinking in a really, really broad sense of what it looks like to understand the very complicated ecological, biotic, abiotic relationships that go on and go into our built and shared environments. And, you know, the kind of complexity that is shown in this diagram is kind of how I feel about a lot of ecological work, and I think how a lot of people feel about it, and it feels almost like there's no place to start because it's such a messy system. But it's these types of pictures are the kinds of the kind of work that I'm interested in, and figuring out how do we get tools to make a lot of these like lines more and more clear. And so the main influences on the research that I do now happen to align with the people who are going to be like judging me for whether or not my thesis is good enough, because they're on my thesis committee. The first one is Joe Paradiso, who is my main advisor from the Media Lab. And there's a project in particular in our group, Responsive Environments, which is the Tidmarsh Living Observatory, which is a cranberry bog that's returning back to nature. And it's fully sensored out. So there's tons of sensor nodes, which you can see through all those little dots. And this is the whole cranberry bog environment. And it's really, really amazing. And it's kind of cool to go in waders and go and sort of check in on all of the electronics. And there's audio streams and there's video streams, and a whole now sort of ecosystem of Massachusetts-based researchers who are basically taking a lot of this environmental data and figuring out what can we do with it. How do we understand what's going on in the changing of the environments? And I love this project, and I also recognize the fact that there's no real way that we can do this for biological data, which was kind of sad for me. And so one of the impetuses for me being in the research group is thinking about how do we get better biological sensors that can maybe not exactly act as these sensors do that are more chemical, physical, audio, video, but sort of get closer to figuring out how we can integrate biology. And then also Professor Jim Collins is my co-advisor in the biological engineering department, and two projects in particular from his lab that I got really excited about before I ended up joining. On the left right here is the BioBits project, which was led by Ali Hong, who graduated from the group. And that was thinking about how do we do better synthetic biology education. And so it was taking synthetic biology out of the lab, not necessarily in a monitoring context, but in an educational context, and a lot of the low cost readers and the low cost tools that you can use to try and create a better understanding of this type of work in the outside of lab context was super exciting to me. And then on the right is CRISPR Biosensors. So there's a CRISPR biomaterials paper that came out maybe a year or two ago, which created all these different new form factors for biosensors. And there were a few of them, and reach out to me if you want more information on the paper. But it had these really nice different ways of figuring out how we can have these CRISPR biosensors interface with the shared environment, and with people, and giving interesting outputs. And so that was really exciting. And then the last thing is this concept of just good enough data. So one of the third advisor is Professor Jennifer Gabrice, who's at the University of Cambridge in the UK. And she has done a lot of work on not biological sensors, but electronic sensors and people, and how we do better participatory design research. She's a sociologist. And so from a sociological perspective, what does it look like to take these sensors outside of the laboratory? And something that I really needed to be reminded of, and especially in a place like MIT, where we try and do some of the most cutting edge research in the world, is that the data doesn't have to be the best when you're taking it outside the lab, and it kind of can't be, because you're taking it outside of ideal circumstances. And so figuring out what it means to have low cost instruments, low tech instruments that people can use, but also figuring out how to integrate someone's personal experiences and how to integrate someone's lived experience as part of the sensing sort of system that you're creating, in order to generate this unique data set that requires person and sensor together, and maybe isn't going to be hitting the limit of detection that you're hoping for, but will be the right the right amount of sensitivity for the data to take you to the next step, like talking to a regulator, or contacting your local water authority, or deciding that you want to look into an issue more. So it doesn't have to be the best data, it just has to be good enough to get you to the next step. And so with all those things in mind, I realized that, again, biological sensing isn't quite there, which was sort of upsetting. This is a picture from SENS, which was the Center for Embedded Networking Systems, which was a project that was run out of California for about 10 years. And it was this huge sensor project, similar to sort of what Tidmarsh is now, where they deployed lots of sensors. And you can see here that there's remote sensing, static, robotic, and handheld. They all have trade-offs for resolution in terms of time and space. But the problem is that there's really no good biological sensors that will give you like any amount of information. And I should also clarify that when I say biological sensors, I mean using biology to sense biology. So there's a lot of biosensors that use biological inputs to get electronic outputs, and that counts as a biosensor. And I also think about using biological input to get a biological output, for instance, getting like a protein made or fluorescence signal or some chemical output. Those are all parts of it. So there are some electronic sensors that can try and estimate biological outputs, but there's no great, really robust biological system that's really quick, really robust, and can sort of sit out for very long periods of time. And so, coming back to this image 10 years later and 15 years later and realizing that we're not super far along was a little disheartening, but also, motivating. And also thinking about just the complexity that goes into environmental research as being one of the real reasons why this is so difficult to do. Like we can't really standardize biological sensing until we standardize as many of these sort of nodes that go into the data infrastructure of environmental sensing, which goes in, especially when you're incorporating people, I guess, to be clear. If you're doing experiments as one person in a lab, you have this incredibly controlled environment. But when you want to take these experiments out of the lab and you want them to be done by people who are not trained the way that you are trained, there are questions in standardization that come into play, both in terms of the methods people are using, the kinds of data, the way that you're collecting the materials, where you're collecting them, how you're measuring them. All those things are super critical in order to make sure you have a cohesive data set that can then be useful. And so the more you can standardize, the better, but these are all things that are quite difficult to standardize when you're incorporating people into the process. And so just to sort of situate myself within the kind of work that I do, I am currently working on sensing systems for CRISPR-based biosensing. And so I actually worked with Cas12a, which is the specific CRISPR protein that we use for our sensing when I was doing my gene drive work. So it was quite a seamless transition to go from that to this, because the enzymes were the same, the types of design I was doing were the same. It's just that instead of waiting months for an animal model to be born and characterized, I can get responses in the order of like minutes to hours, which was quite exciting to have that kind of switch. But in specific, I do this CRISPR-based sensing system. So it's a cell-free system, as you can see from this chart. This is a paper that just came out recently, and it is a very nice comprehensive look at what it looks like for synthetic biology to be doing field-based work, and also obviously assessing the fact that it hasn't quite been done to the extent that we really need it to. But the system that I work with is, to me, ideal because it's a cell-free system. And so I don't have to worry about the types of sort of rules and regulations and containment that is involved in things that are living cell systems, because those are, I guess, classified more in a GMO context, and then you get into a lot of regulation that you maybe don't really want to get started with. But just to zoom out a bit, so Cas12a is an enzyme that was discovered a few years ago. Cas9 is the most well-known CRISPR enzyme, I would say, but Cas12a is great in its own special way. And so it has the enzyme and then the guide that it complexes to. And the way that it works for the specific use case that I have is that, so Cas12a has this unique off-target property, which is that once it binds to the DNA it's looking for, so the guide RNA tells it where to go, and once it goes to that place, if it finds that piece of DNA, it will cut the DNA, but it will also start shredding any single-stranded DNA in the vicinity of that double-stranded DNA. And so people have basically utilized this as a way of doing a really robust and quick biological sensor. So you first need to have a nucleic acid input. So there's two different systems. Sherlock was first published by Feng Zhang and Jim Collins' lab in actually the same issue, I believe, that Detector was published, which is the Cas. So Sherlock is Cas13, which is RNA sensing. Detector is Cas12, and that is DNA sensing. And Cas12, the Detector system, was published by the Doudna lab. And so depending on your nucleic acid input, you choose whether you have double-stranded DNA or RNA, and then you do this isothermal amplification step. And so that is quite important when you want to get to be really, really sensitive. So by itself, the CRISPR enzyme can detect something in the picomolar range. But when you do isothermal amplification, you basically try and amplify the specific target you're looking for, you can go down to attomolar detection, which is 10 to the -18th, which is super sensitive, which is great, especially for environmental samples where things are really messy or dilute. It's really hard to get as much, DNA material as you might want. And once it amplifies your target, it goes through this detection system where basically you fill the reaction with these single-stranded blocked fluorescence molecules. So once the Cas12a finds its target and it cuts the target, it starts shredding everything in vicinity that's single-stranded, and that includes these single-stranded blocked fluorescence molecules. And so if you find the target, you'll start getting this fluorescence signal because the fluorescence only will show if it's getting shredded. And it's super quick, super robust, which is very exciting. So this is kind of my workflow. I do mostly waterborne pathogen sensing, and that's the sort of environmental space that I'm quite interested in. So you would, in theory, collect a water sample, you would maybe filter it to try and concentrate the thing that you're looking for. And then you can actually do the last 3 steps all at once. So you can do a one-pot reaction that has the amplification and the CRISPR detection, and you just wait for a fluorescence signal to come out. And the pros of this specific system, I'm obviously quite biased, but it's one of the most rapid. It's quite rapid for what it is because there's no sort of transcription translation needed. So there are other types of systems you can use for biological sensing that have been published in previously, things like toeholds and these cell-free transcription translation systems. So waiting for translation, it takes quite a bit of time. So in this case, you're just basically waiting for the fluorescence to show up. So that can happen on the order of minutes. You can get single copy detection of the thing you're looking for. And the reaction runs at 37C, which is not quite room temperature. It actually, no, the amplification can run at room temperature, but the CRISPR sensing not very well. But 37 is a reasonable temperature to try and get at if you have to do a sort of like DIY system. It's sequence specific. So for a lot of the microbial analysis on the environmental scale, thinking about like not just looking for a specific organism, but actually like specific toxins and things like that is to me quite important. So that's great. But the downside is they're not reusable, and sometimes when you get to low concentrations, it's a bit unreliable. But moving through, so this is a project that this is like my first attempt at thinking about taking these projects out of the lab in the sort of specific CRISPR biosensor instantiation. So this is a tube version, not a paper version. The paper version is coming, but I can't show any of those experiments right now. But this is a tiny little reader, and it has on the side a lysis chamber, which basically heats up to inactivate any of the things that would be inside your sample of interest. This is specific for saliva, but usually I would make it more water detection. And then you basically move over the little filter. So these have little filters. You move the filter over and you plunge it through into this freeze-dried reaction. And then you have these built-in LEDs that do fluorescence reading while the temperature is staying at 37 to incubate the reaction. So this is sort of like most minimal lab possible. This thing is, like less than $10 to make if you have a 3D printer, which obviously people do not always have 3D printers. So that's a bit of an issue. And this is sort of thinking about like, what does it look like to make a tiny lab? And I've spent a lot of time actually in specific working with libraries and librarians, and there are so many actual like library resources for Fab Labs that are developing and specific grants that are going out to libraries to figure out how do we increase the capacity of libraries to sort of be maker spaces as well. And I think that's really important when I think about getting access to shops. It feels like it's something that's just on an academic level and it's not accessible to the public, but actually there is a lot of really exciting work that's being done on a community scale for specific cities, not just cities like Cambridge, but like all over the US, figuring out how can we build capacity, but also how can we think about maker spaces as community resources. So even though not everyone has a 3D printer, you don't necessarily need a 3D printer in your house in order to be able to get access to those tools to make these things, which is, I think, something that's important to clarify. But there's a larger question, at least for me, which is I read papers all the time, feels like that's my job. And everything talks about being low cost and deployable and low resource and field ready, but none of them ever actually deploy. There's like little to no research on any of these biological systems ever being activated in an out of lab context. So you have all this really beautiful, perfect lab data that looks amazing, and you have nothing to benchmark it to other than someone telling you that in theory the cost can be quite low or the tools can be used. And so one of the reasons why these things haven't really been deployed is that a lot of the times it requires specialized or custom equipment. So for instance, my reader, that's great, but that's not really going to be helpful to somebody unless they have the time and interest in order to build that type of system. Typically, biosensors are not reusable. A lot of electronic sensors, which I've done a lot of community work with electronic sensors, it's a lot easier to do electronic sensors because those can last for quite a while. So some of them need to be calibrated every time. Some can be calibrated only, once every year, but they can put it out and keep getting continuous information. Whereas with biological systems, you can't actually do that because it's a chemical reaction or a biological reaction, and once it sort of runs out, that's it, and you have to start again. Biology just is not as reproducible as things like electronic sensors. That's something that people are working on all the time, and it's obviously an area of optimization, but it's still something that people need to work much more on. And sometimes there are issues with low quantities, which I think is mostly because of the messy sample issue. So in specific for environmental monitoring, but also true for diagnostics and biological monitoring, things like blood and saliva and water and soil and all that kind of stuff are quite messy. And a lot of biology doesn't work as well when you introduce a messy sample. But for biological sensors in specific, if you are required to do really intense sample preparation before you end up doing the actual sensing, you're going to need a lab before you even get started. And a lot of the biosensing papers have these little tricks that you'll basically do an entire very complicated lab cleanup before you run it on your low cost device, which are just not things you can sort of export to out of lab context. Some require special approval. So in specific, when you're using living organisms, you might not be able to deploy just because of the fact that you are trying to put a GMO in a country that wouldn't allow you to do it, or in a place where it wouldn't allow you. You might need local, state, federal approval for the kind of work you want to do. Sometimes, these are even seen as bioweapons, which is kind of crazy, but something to be aware of. And then the actual reasons outside of the technical ones are just that researchers don't really have the time. So you might be a biological engineer or a biologist and you want to do this work, but you have other things that are pulling at you, which are your direct research requirements, and you just might not have the time in order to take the project to the next step once you publish it, because the publisher doesn't require you to do that. We don't have the skills. So a lot of the work with community and specific, it's a whole separate skill set. And for me, it was something that I was already interested in. So it was quite exciting. But if it's not something that's in your interest or it's not in your wheelhouse, it's just really, really difficult. It just takes a completely different set of skills, a completely different sort of mindset to accomplish. And because we don't have to, it's totally allowed within the field of biological engineering to publish that something is field deployable that has never been deployed, and there's no proof that that's actually true. And so I think that's also a really important thing to recognize, and something that I think everyone who works within this space of low cost tech is kind of complicit in, is knowing the work that you're doing isn't going to be as deployable as you want it to be, but not really having any not having anything else to do about it. So I guess thinking about what it takes to get these biosensors out into the environment, and also why do we even want to do that? And this is just more of the philosophical side of the talk. I have some pictures of the kind of community work that I've done, and I'll talk a bit about it, but I'm more interested in hearing from the other people within the club, and also from maybe people who are listening to the talk, what you're thinking. First of all, I think that it's really important to deploy them because that's what we say that they're meant to be designed for. So there's there are some good reasons for trying to make these really interesting low-cost systems in the laboratory, but if you're not ever going to take it out of the lab, you have really expensive systems that can probably work a lot better. And so if it's just to to say you can do it or to publish it, that's one thing, but to actually do something with it is a whole nother thing. So that I think is a really critical component, and something that should really be emphasized for especially labs that do a lot of these low cost design and deployment. And I have a bunch of friends who really do go out and test their technology, and it's really exciting, and it's a whole nother like learning experience. But more importantly is that communities can use these tools. And so people are doing amazing work all the time in laboratories, and there's so much benefit that can happen. And also so much redundancy that goes on when you don't actually realize that you're all solving the same problems. And so figuring out how to get these tools out into the world, how to get people using them, how to get people interpreting them, it's just so much better. So there's I work with two communities, one that's local, so the Mystic River Watershed Association, which I don't have any pictures for because I've been doing most of my work with them currently virtual, and I feel like putting screenshots of Zoom is like not what people need to see while they're on a Zoom. But that's okay. So the community that I was working with before the pandemic hit, and I had so MIT shut down travel on a Thursday. I was supposed to fly out again that Monday and then had to cancel everything. And I don't think I'm allowed back in the country until everyone in New Zealand is vaccinated. So that will be some time because I don't know if they've started their vaccine rollout yet. But there's this tiny community. It's a town of like 700. I met them through a contact I had at the EPA New Zealand. So when I was doing my gene drive work, we had been working with New Zealand, who has a terrible invasive rodent problem, and just working, speaking with communities and asking them questions. I think it's illegal to have any GMO in New Zealand right now, from the last I checked. And so it wasn't as if we were going to be putting gene drives inside New Zealand, but it was interesting to get sort of input from people who are completely on the other spectrum, but really in need of a solution to their invasive pest issue that's ideally not like chemical related. And the contact from the EPA, I'd reached out to them because I was interested in figuring out how I can learn more from communities who are doing their own local monitoring. And he connected me with this community where actually his father-in-law lives and is retired in. And it's this lovely community. And I think something that I think a lot about when I think about community engagement in particular, and thinking about taking these projects outside of the lab, is just that, taking this sensing of like biological sensing and bringing it to a new sort of level of also sensing with your body and sensing through being physically present in a place, it just changes the stakes that are present within the kind of work that you're doing, and it makes it feel so much more real. I think a lot of reasons why people become engineers, and a lot of maybe people on the call are thinking about engineering, is because you like want to do something, and engineers are quite application focused. And I think when you get to go to communities and you get to see the places where you want to be working, it just gives you a whole nother set of understanding of what you're doing and why you're doing it, which I think is really wonderful. This is some of the community members, in particular, Sophie, who's standing up. She just passed away this past spring. And I saw this picture of her and I got so happy. But the way that I usually do my workshops in this community in particular, but also in general, is if I'm working with a community and they're really interested in testing out tools, the first thing that you do obviously is thinking about how the community conceives of their own environment and the issues that they think are most important. And so usually the whole morning is reserved just for the community participants who talk about the issues that they have, historical experiences with the place. And so in particular for the community in Mahia, it's great because whenever we have workshops, it's always quite intergenerational, and there's usually at least 3 generations present. And so to get a chance to listen to the older generations talk about what let's say the water looked like when they were kids, what they heard from their grandparents about the area, which is really special because not everyone feels so tied to a place, but working with Indigenous communities in particular, the connection to place is really critical. If there isn't an option to just go somewhere else, but also because the relationship is so critical and so intimate, there's such a long history that's present that you can be learning from. And that data is so much more important than whatever you're going to collect with your sensor on that day. And so that is sort of the structure. Is the morning is mostly just for the community, and then we'll usually have a lunch break, and then the afternoon is for the water analysis. When I do workshops with children, usually we also do the same thing where we'll do the morning as discussing, we'll have a snack break, and then we'll do the the application. And I think it's important to keep both of those together as much as possible. Spreading it out over the course of days is good, but trying to have direct applications to what you're talking about, I think just like helps things stick in people's brains a bit better. And another really great thing about working with Indigenous communities, but also just local communities in general that are just people who live in places, is thinking about using, lots of different parts of yourself within the research process. I think this is a quote from Robin Molkimmer from Breeding Sweetgrass, which is a book that I think most people who are interested in thinking about native science think about, and thinking about what it means to bring, like, your mind and your body, your emotion, your spirit all into research. To say that at MIT, it's like, oh, you care about people, but I think it's really important, and I think it's something that gets lost within some of the scientific work, is just to remember, like, you're also just like a human being in this place at this time, and how do you embrace that instead of trying to compensate with technology, which, technology isn't meant to compensate, it's meant to sort of complement. So figuring out how to make that happen. And so this is just some nice pictures of different sampling that we do. So we go out into the reefs itself and collect water, and the way that we choose where we go to sample is based on the conversations we have in the morning. And I think that's another really important thing when it comes to doing environmental sensing with the community, is that there is no way, going back to sort of the electronic sensing, there's no way currently to sort of deploy the biosensors in the way that I conceive them, in a way that's like sort of autonomous. And thinking about where you want to sample is quite important. So it's too much information, and it's too much maybe not super useful information to just deploy everywhere all at once. And so you really have to start making choices about what you want to be looking at, and when and where and how, and what that data point is going to look like. And so having the communities be the ones who direct you to the locations, or having communities decide for themselves places that they think are most important, is really critical, because you can't sense everywhere. And so having community input and community guidance and community direction on where the places are that actually need to be monitored, and whether it's because somebody said, actually this place used to have studs in it all the time, or we keep seeing that there's like sewage that ends up here, or like this place has a weird smell. Like, all of those pieces of information are really important within the sort of whole of the story of the kind of sensing that you're doing on the biological scale. And those are all biological indicators that would be an interesting place to sense. And also just thinking about the idea of like what it means to restore relationships, and thinking about how like our relationship with the land and with environment is like very intimate, I would say. And using sensing as a way of restoring a relationship, I think, is quite important and quite beautiful, and figuring out how we can understand the language of the environment better on a lot of different levels will help us as we try and adapt, as the future brings a lot of changes within our natural environments. And so the more we can understand nature, understand biology, and build and strengthen that relationship, the better we can restore it, but also the better we can sort of navigate that relationship once things change, which they're always changing, but they will start to change, and are changing more rapidly now than they have in the past. And there's been a lot of different types of sensing. I've done biological, chemical, electronic sensing, and I have a lot of recommendations for people if you're interested in like what works best and how to sort of activate people's interest. People love light up things. This is a sensor that was originally made by my friend Laura Perovich, and it was kind of my introduction into soldering at a time when I decided I wanted to gain more skills within electronic sensing. And these are really cool little water sensors that when they go into the water and sense for this, there was one for conductivity and one for temperature, they start changing colors. And so talking with the community of people who maybe don't have a super strong science background and explaining something like a color change, it's an output that's quite intuitive. And I think thinking about intuition within outputs of biological systems are usually not so important if you're a scientist, but quite important when you're a community member. So to have a system that's really intuitive in its output was just like a super great and easy thing to talk about. And so thinking about engagement projects and how you design it, these are some rules that come from the book Decolonizing Methodologies, which is by Linda, and she lays out a lot of really important things. These are sort of based on, so the community that I work with in New Zealand is a Maori community, so the indigenous Polynesian people of the land. And Linda Tuiwai Smith, who's the one who wrote this book, is also Maori, and thinking about how you incorporate indigenous methodologies into research itself. And I think a lot of these questions that she asks are really important, even just broadly for community engagement, regardless of whether you're working with an indigenous community or not. And thinking about, I guess at the heart of this is the kind of power dynamics of the relationship, but also the give and take of the relationship. So thinking about defining the research problems, why it matters to study the kind of knowledge that's gained on either side, how to limit the amount of negative outputs that happen within a project, the accountability. So I think a lot of times when you're doing community engagement for students, there's a lot of turnover. And so thinking about who the point person is, how that transfers, what that relationship building looks like over time. And then the idea of just like, so what's the process for supporting the researchers, but also supporting the community and supporting the project. And that could be support on a emotional level, it could be on a financial level, and it could be on a technical level. Like all those things are quite important. And I have a very long list of like things to do if you want to work with the community. If people want to ask me about it, I'm happy to share it, and you can send me an email. But I guess this is the last slide. But in specific, thinking about environmental, biological sensing, thinking about working with communities. I think to change the mindset from like not just developing the straight data sets, but also like stories around the data. And that sort of goes back to the just good enough data. It's like, how do we contextualize this kind of work that we want to be doing when you're doing community work? How do you incorporate the stories of the people who you're working with and who have all this lived experience and who are basically human sensors? And also embracing the fact that the data is saying data diversity is kind of like a nice way of saying that the data is kind of messy. But thinking about, what it looks like to have different types of data. So you might think the specific biological output you get is like, doesn't look very great, it's kind of crummy. But when you contextualize it within all these different other types of data, it's a lot more interesting than just, having a validation experiment in a lab. And then also just thinking about like leaning into what it looks like to do interdisciplinary research. And at MIT, I feel like that is sometimes talked about and sometimes not, depending on the kind of like context that you're in. But I'm from the media lab, and all the work that we do is like inherently not in any discipline at all, which can be sometimes really hard, but is great. And I purposely chose to be in the media lab because I wanted to do this work that's kind of between fields, and I wanted to work with sociologists and biologists and electronic sensor people, and it is really exciting, but it kind of requires you to be okay with the fact that the research is going to be not exactly what you expected. Yeah, and I wanted to thank the people. Oh, sorry, I do a final slide. We need to take out we need to take biosensors out of the lab because we also just really need help on environmental monitoring level. There's so much we have to see that we should be watching that we just can't. We don't have the tools for it. And we've started developing them in the lab in the past few years. And so figuring out how we can more quickly get them out into the world, even just from a test capacity. So it doesn't have to be a product that's like, ready made and on the market, but there's so little just sort of field testing, pilot testing, research that's done, and figuring out how to do that as soon as possible will allow us to hopefully have so many more people who are involved in sensing, who know the place better than any scientist ever could, and who can be gathering their own research, deciding for themselves what they want to do with their own environments, their own ecosystems. And hopefully we will benefit. So that's what I have. I'm sorry if it was too long, but thank you so much for having me, and feel free to reach out if there's any questions.
Oh, that was beautiful. Like, I felt that hearing about your interactions with like actual communities really grounds the work that you're doing. Right now, I'm contributing to research that also is doing open source devices, and I just kept hearing about the same problems that we have. I mean, I have some questions, but but I would like to open it up to the chat. Owen Letty has a question. So Actually, does Owen want to ask this question directly? I can allow you to unmute right now. You have quite a set of questions. You have the floor.
Hi, sorry. Thank you for the amazing talk. I just wanted to ask you about how once you've created this kind of biosensor and deployed it in a community setting, how do you go about making the kind of data you collect broadly accessible and interpretable? When I was in high school, I was involved in an effort that was sort of tangentially connected to Safecast, which I know was affiliated with the Media Lab, looking at radiation monitoring and water quality monitoring. And I wonder if you think the kind of integrated online sort of data portal that Safecast created is a good model, or how it could be improved upon, or how it could be adapted for the kind of project that you're working on?
That's a great question. Yeah, in the like long flow chart of things, maybe I'll share just so I can show that. In the sort of flow chart, let's see. So there is like an area for outputs. I actually don't even know where it is. Maybe I'm not going to show this then. But yeah, I think there's a huge question when it comes to the kind of data that's produced, and I think it's actually community dependent. So for some communities, putting it online is great and super useful. For some, most people don't have computers, so don't have an interest. Some people don't even trust putting information online, or don't really want these types of data to be held on university servers and things like that. So I've experienced like the full range. Definitely the communities in Massachusetts are a lot more like pro putting things online than the communities I work with in New Zealand. And I think that has to do a lot with mistrust of government. And I think that's always really important to think about when you're thinking about projects. And not everybody has the same relationship with, the people around them in the same way. So I think it really depends. I think for some communities, having a web page that you can point to or land to, or putting out an exhibition in the community, whether it's like an art exhibition or some sort of art science hybrid exhibition, they're all really valid and really interesting ways. And I think it just really depends. Yeah, and it also depends on the kind of information and whether it's like really critical for the safety of people. So for Safecast and like radiation and things like that, I think they wanted to get it as well known as possible or out to the public, and it felt like a necessity. But for some of this stuff, if you're monitoring, for instance, in New Zealand, sometimes we just monitor really critical species that are going extinct. So when you're doing that kind of sort of biosensing that's related to things that are just having to do with, let's say, organism count, you don't really need to publicize that in the same way that if you're trying to publicize the fact that the drinking water is contaminated. But it certainly, it is complicated because it is very community dependent. And I think it's really critical from the beginning of the project to be thinking about what those outputs are going to look like, and figuring out how to like sort of develop that in a way. I don't know if that's really an answer, but it's just like an important thing to think about, and it will change regardless of the community. But I think personally, I've done exhibitions, and exhibitions have been really nice and really great ways of both giving back to the community and engaging with the public in a broader sense, like on a community level. And there's also sort of like archiving that you can do for that. And I also love libraries, and I think libraries are great community archival resources. And so figuring out whether or not it's like putting the data in a library, putting the data in an archive, putting the data on a computer, which is also, I guess, an archive. It varies.
Great, thank you so much.
I sort of have like a policy related question, if you're up for that.
Yeah, sure.
So right now I've been like, I'm not familiar with the reasons why there are so, like when we look at like the GMO policies that are out there, like GMO related regulations seem to get in the way of a lot of like open source biology efforts. Do you think that we should like work towards destigmatizing GMOs, or like actually take into account like maybe community feelings and anxieties about them and just work with the policies in place?
I think both. I think at the same time as you're trying to figure out ways of destigmatizing GMOs, there are also like really, really valid concerns that underlie those things. And I think there's a lot of, like, you can sort of learn the sort of politics side of why GMOs are so problematic in terms of like a global context and how they've been used as something to be very concerned about. And I think also what's difficult is that you can only reassure people so much, especially if you're working within research, because you're only working in a lab context. And so I think it's important to have conversations about it. I think it's important to, not come in telling them like, I know best, and this is what GMOs are, and this is why you shouldn't be concerned. But I think to have a really robust conversation about why they are concerned, about what are the things that they've heard. It's really interesting because I've like also been like across the spectrum. Like I've had people tell me that, the government is like currently hacking our children's brains and they're taking over, and the UN is a shell corporation of these people who are trying to like kill 95% of the world. And I've also worked with people who are like, I would love GMOs in everything that I use, and everything in between. And I think it's hard to be, I don't think you have to be impartial. I think you can have a perspective on it, but I think also to, if you're doing community work and it's going to be a long-term relationship, for you to figure out how to have conversations and understanding, where people are getting their information from, who are the people that they trust, why are there already reasons why they don't trust these specific things, what's going into those conversations. Like that's important to know contextually. And then in terms of actually dealing with regulation, I will say rarely is the community that you're working with the people who are probably going to be the like official regulators, on a governmental level. And so I think it's cool to have two hats and think about what it means to do like really interesting policy in a lot of the GMO issues. It's like international policy around these types of areas. But I think it's also just like, important to understand the community's relationship to these issues. And obviously you can get approval from the government, but you don't get approval from the community. And so it's like still unethical for you to be doing something. And so you kind of need both hard and soft approval. And to me, the soft approval is the hard approval. So it's difficult, but it's also, it's just like really important foundationally. And also I will say there's like a third thing to this, which is that, the idea of being like the science savior is like problematic, of course, but also there are a lot of communities that you wouldn't expect them to be so interested to be listening to scientists, but there's a lot of communities where like the power imbalance is such that like even if you're coming in as a student, they might just like sort of take everything that you say as like being the law, and they put a lot of belief in you, because you can be faced with a lot of skepticism, with a lot of just sort of like blind faith. And I think to be considering the kind of like position of power that you hold within that arena and trying to be very careful about that is really important. And I think to just convince someone to change their ways because you think you know better, like that's not the point of doing engagement. That's not like engagement. And so trying to make sure that you're like also learning from the people around you and contributing what you can. I guess this gets more into like participatory action research, but thinking about having a more horizontal power dynamic, and obviously being there for the expertise that you're there for. So you are there because your expertise is being someone who does Western science. So it's like, that's the whole point of you being there, is to contribute that, but you don't want that to sort of overwhelm the other things.
Yeah, I really appreciate your perspective, because I feel like my own intuition is to want to centralize everything, and that kind of excludes a lot of different opinions about the work that you're doing. So it is 4:30. I'm going to drop some like general information about Biomakers, but I know that you mentioned that people can reach out to you via email. If you want to drop your email in the chat to our panelists and attendees chat, feel free to do that. Great.
Oh, that was just a panelist shoot. Sorry, I'll do it to everyone. Yeah, thank you so much for having me. I feel like every time I speak about this, it always feels like it's insufficient. And sometimes it's easier when you have like more constrained or specific things to talk about, but feel free to ask questions. And if you're interested in the research, like we're both research groups like potentially you could be interested in like getting Europs if you want to do these types of projects, not necessarily with me, but just like in general. And I wish you all the best.
Awesome.
Great. Thank you so
Again,
much.
thanks for coming.
Thanks, guys.
Thank
Have
you
a
so
great day.
much.